Hey, this is our final evidence, and we're going to be talking about topic 11, which is about the nervous system. We're the team conformed by Alicia Lugo Salcedo and Margarita Maceo Calleros. An overview of the nervous system. The nervous system is divided into the central nervous system, the CNS, which consists of the brain and spinal cord, and the peripheral nervous system, the PNS, which is made of nerves and ganglia. The nervous system helps to maintain homeostasis and integrates all body activities by sensing changes in the internal and external environments, the sensory function, interpreting them, the integrative function, and reacting to them, the motor function. Types of neurons. The nervous system consists mainly of cells called neurons, and there are three types. The sensory neurons, which carry sensory information from the cranial and spinal nerves into the CNS. The interneurons, which are found within the CNS and receive sensory information and pass it through other interneurons or to motor neurons. The motor neurons, which carry information from the CNS through the nerves to effectors such as the muscles or glands. Here's a visual representation of the three types of neurons. As you can see, at the start there's a sensory neuron, which has dendrites in a direction of conduction. Then it goes into a cell body and passes through an axon in order to reach another cell body in the interneuron. Then it goes through another axon, which leads to a cell body that passes through another axon, and finally reaches the motor neuron. Within the neurons, there are concepts such as nerve impulse. The nervous system uses a nerve impulse so it can convey information along the neuron. The nerve impulse, it is an action potential. When a neuron is not firing, it is said to be an arresting potential, but when the neuron is firing, it is carrying out an action potential. Resting potential. During resting potential, the inside of the axon is negative compared to the outside. The sodium ion concentration is greater outside of the axon, while the potassium ion concentration is greater inside of the axon. Now, the distribution of ions is maintained by the action of carrier proteins in the membrane called sodium-potassium pumps, which actively transport sodium ions out of and potassium ions into the axon. An action potential is a rapid change in polarity across the axon membrane as the nerve impulse occurs. If the stimulus causes the axon membrane to depolarize to a certain level called the threshold, then an action potential occurs. It requires two types of gated channels in the membrane. One gated channel allows sodium ions to pass through the membrane to the inside of the axon, while the second gated channel allows potassium ions to pass through the membrane to the outside of the cell. The polarization occurs when the action potential begins and the sodium channels open first, allowing sodium ions to enter the axon. This is called depolarization because the change inside the axon went from negative to positive. Repolarization happens when the potassium channels open, allowing potassium ions to flow out of the axon. This is called repolarization because the inside of the axon returns to a negative charge. Within the axons, there are concepts such as refractory period, in which the action potential travels down the axon one small section at a time in a non-myelinated axon. As soon as the action potential has moved on, the previous section suffers a refractory period during which sodium ions channels cannot open, resulting in the forward movement of the action potential. During the refractory period, the membrane potential is more negative than resting potential or hyperpolarized. When the refractory period is over, the sodium-potassium pumps sodium back out of the axon and potassium back in, returning the axon to resting potential. Myelinated axons. In myelinated axons, the gated ion channels that produce action potential are concentrated at the nodes of Ramier. The action potential then jumps from one node to another, allowing the action potential to travel faster down the axon. Chemical synapse. Every axon branches into many swollen endings called axon terminals, which are in close contact to either the cell body or dendrite of another neuron. This region is of close proximity between the axon terminal of one neuron and the cell body or dendrite of another is called chemical synapse. Synaptic cleft. The two neurons never actually touch one another but are separated by a tiny gap called the synaptic cleft. Postsynaptic membrane. At the synapse, the membrane of the first neuron is called the presynaptic membrane, and the membrane of the second neuron is called the postsynaptic membrane. Chemical synapse. Communication between two neurons occurs at a chemical synapse and is carried out by chemicals called neurotransmitters that are stored in vesicles within the axon terminals. Communication between two neurons occurs at a chemical synapse and is carried out by chemicals called neurotransmitters that are stored in vesicles within the axon terminals. When nerve impulses traveling along an axon reach the axon terminal, neurotransmitters molecules are released into the synaptic cleft. They diffuse across the cleft and bind to specific receptors on the postsynaptic membrane. The response of the postsynaptic neuron depends on the type of neurotransmitter and the type of receptors. The response can be excitatory, causing depolarization, which favors an action potential, or inhibitory, in which it causes hyperpolarization, which results in stopping the action potential from happening. Here we have a visual representation of a chemical synapse. First, we have the postsynaptic neuron, which then goes into an axon presynaptic neuron. Inside, there is a mitochondria, and then there's the axon terminals at the border of it, the synaptic vesicles, which are portrayed as little circles, the synaptic cleft, which is under it, and then the receptor sites, the neurotransmitter, and the postsynaptic membrane. Many different neurotransmitters have been identified. Two well-known neurotransmitters are acetylcholine and norepinephrine. Others include serotonin and dopamine. After the neurotransmitters have been released into the synaptic cleft and has initiated a response, it is removed from the cleft either by inactivating or rapidly reabsorbing the neurotransmitter. The short existence of neurotransmitters at the synapse prevents continuous stimulation or inhibition of postsynaptic neurons. Drugs. Many psychoactive drugs affect the nervous system by either interfering with or potentiating the action of neurotransmitters. Drugs can enhance or block the release of neurotransmitters, mimic the action of a neurotransmitter, or block the receptor or interfere with the removal of a neurotransmitter from the synaptic cleft.
The central and peripheral nervous systems. The peripheral nervous system is located outside the CNS and consists of nerves and ganglia. Nerves are composed of bundles of axons called nerve fibers. Ganglia are collocations of neuron cell bodies located outside of the CNS. The PNS is further divided into the somatic system and the autonomic system. The somatic system innervates the skin, skeletal muscles, and tendons. It consists of nerves that carry sensory information from the sensory receptors to the CNS and motor fibers that carry information from the CNS to the skeletal muscles. The autonomic system of PNS regulates the activity of cardiac muscle, smooth muscle, and glands. The sympathetic division of autonomic system consists of preganglionic neurons whose cell bodies are located in the thoracolumbar region of the spinal cord. The parasympathetic division of the autonomic system contains neurons found in some of the craniosacral region of the autonomic system. It promotes internal responses associated with rest and digest. Acetylcholine is the main neurotransmitter used by the parasympathetic division. Reflex. Reflex is a fast, automatic, and unplanned sequence of actions that occur in response of a particular stimulus. There are types of reflex. Inborn reflexes. An example is putting your hand away from something hot. The spinal reflexes, the integration that takes place in the spinal cord. Cranial reflexes, if integration occurs in the brainstem. And somatic reflexes involve the contraction of the skeletal muscles, while the automatic reflexes involve the response of smooth muscle, cardiac muscle, and glands. Here we have an image of a reflex arc. The number 1 point is the sensory receptor. It's either the dendrite at the distal end of the sensory neuron or associated sensory structure. Number 2 is the sensory neuron. A nerve impulses travel from the sensory receptor along the axon of a sensory neuron towards the axon terminals that are located in the gray matter of the spinal cord or brainstem. The number 3, integrating center. The integrating center is one or more regions of gray matter within the CNS. Number 4, motor neuron. Nerve impulses from the integrating center leave the CNS along motor neurons to effectors. And the last one, number 5, effectors, are parts of the body that respond to the motor nerve impulse, such as a muscle or glands. Physiological basis for depression and happiness. Depression is a mood disorder caused by psychological, biological, and environmental factors. The symptoms of depression are sad and empty feelings, decreased interest in activities, pronounced changes in sleep, appetite, and energy, being agitated, difficulty of thinking, and suicide. Depression neurotransmitters. The serotonin regulates mood, appetite, sensory perception, and other functions. Norepinephrine is involved in alertness, and dopamine regulates movement, emotion, motivation, and feelings of pleasure. Happiness. Happiness is an emotion that brings pleasure and plays a crucial role in our physiological well-being. Dopamine always plays an important role in happiness because it mediates the transfer of positive emotions. Happiness is also associated with lower levels of cortisol and therefore a stronger immune system and less stress.